This is Lecture 8, Development of the Nervous System, and it corresponds with Chapter 8 of your textbook. Feel free to skim the first section. Good to look over and review anything you need to, and then read the rest of the sections as normal.
Alright, so jumping into Chapter 8, Section 8.1, I'm not really going to be covering. I recommend skimming it, but I'm not going to be taking any homework information from it or exam material from it. They do mention that genetics should be conceptualized like a blueprint. This is a concept we had talked about before, but I just want to bring it up as a reminder that genetics are not a complete determinant. They are more just like a blueprint, like when you're making a house and general instructions, but there are other factors that influence the final product. So Section 8.2, the neurobiology of development. In vertebrates, embryos look very similar, both in the body and early brain development across species. So you see this figure down here. There's the embryos of a salamander, a chick, and a human, and you see that they look very similar. So early brain development, very early brain development, in utero looks very similar across vertebrates. So a rough timeline for the gross development of the human nervous system. During weeks 1 to 2, so the first 14 days. Day 1, of course, the sperm joins the egg, and this is fertilization. This creates a single cell human, which is called the zygote, and these cells divide, starting on day 2. Continue division until around day 15, when they form this flat plate called the embryonic disc, flat plate with a little raise in the middle. Weeks 2 to 8, we have an embryo, this sheet of cells with the raised middle. Week 3, the neural plate, which is this raised bit in the middle, folds and curves to form the neural tube. And the neural tube is extremely important structure from which the brain and the spinal cord develop. So it's literally just this sheet of tissue that folds and curls around to make this little tube. And from that tube, all of the cells are dividing and being created, and this will eventually be the brain and the spinal cord. Week 7, the embryo starts to look like a little person. Week 11, the brain starts to look humanoid, so it starts to look like that human brain that we know. And then by the 9th month, the brain starts to look like an adult human brain, but the cellular structure is a bit different. So the origins of neurons and glia. The neural tube is the origin of all brain cells, as I mentioned before. Neural stem cells are cells that can self-renew. So neural stem cells can create many different types of cells. In fact, all of the cells in the central nervous system are created from neural stem cells, and this includes both neurons and glial cells. For adult humans, our ventricles, which if you remember are those spaces in the brain filled with cerebral spinal fluid, are aligned with neural stem cells, and this area is called the subventricular zone. Neural stem cells create progenitor cells, and which in turn create neuroblasts and glioblasts, which are the precursors to neurons and glial cells. And neuroblasts and glioblasts can create all the different types of neurons and all the different types of glial cells. So cells differentiate, they turn into the different types of neurons and glial cells based on what genes are expressed or turned on. Cells that are turned on will make, or genes that are turned on will make specific proteins, which produces this differentiation in the cells. One process by which this happens is gene methylation. So there's a compound called methyl groups, and they bind to and suppress a base pair in DNA, in genes. And this is one way in which genes are expressed. So some genes are turned off, which expresses other genes. This is also one of the main mechanisms of epigenetics, or passing on characteristics that are gained in life, passing those on to offspring. Cell differentiation is regulated by a series of chemical signals, and these are called neurotropic factors. So neuronal growth and development, after neuroblasts and glioblasts are formed, they migrate to the correct area of the brain. So some cells will migrate to the frontal lobe or frontal area. Some will migrate back to the visual cortex area. at which point they differentiate and grow dendrites and axons. Neurogenesis is mostly finished at about 5 months after conception, except in the hippocampus. In the hippocampus, new neurons are actually developed throughout the entire lifespan, so it's kind of a special part of the brain. If you remember, the hippocampus is responsible for a lot of the memory functions in the brain, so you can see why we would need new neurons in that area of the brain to help form new memories. And the creation of new glial cells continues throughout the life. So unlike neurons, where not many new neurons are being produced throughout adult human life, new glial cells are continuing to be formed throughout adult human life. Cell migration starts shortly after neurogenesis, so shortly after those neuroblasts and glioblasts are created. Cell migration takes about 6 weeks, and after which time differentiation happens. The cells are differentiated into different types of neurons. After the cells are differentiated and become the specific types of neurons, they then mature, which means that they form dendrites and axons, synapses between the neurons. And neuronal maturation can continue through adulthood in certain areas of the brain. Also remember that the cortex is organized in layers. So you remember there were like 6 layers in many areas of the cortex. The subventricular zone functions as a sort of map that sends cells to different areas of the brain. So if a new neuron is in a certain part of the subventricular zone, it's going to be sent to kind of a corresponding part of the brain. Radial glial cells are special glial cells that create kind of a pathway, sort of create a bit of a rope that neurons follow to the correct layer of the cortex. Some cortical neurons do follow chemical signals instead of these radial glial cells, but most of the migration happens through radial glial cells. And cortical layers develop from the inside out. So it's just like building a house. You would build the foundation first, and then the walls, and then the roof. The cortical layers are built the same way. So for neuronal maturation, first there's dendrite development, and this involves dendritic arborization, arbor being like tree. So this just means like the branching of the dendrites. And then spine growth, or these little like nubs that are created on the dendritic branches. And then there's axonal development, which compared to dendritic development is very fast. Axons, if you remember, can go really far, very far through the body. So there are some axons that go all the way from the central nervous system down to your big toe in your foot. Axons take cues from the environment, from the body, to reach the target. Simple synapses form in the brain 5 months after conception, and synapses start to increase very quickly after birth. So after the baby has been born, they're suddenly exposed to this whole world of sensory stimuli and experiences that they didn't have inside the mother's womb. So they're quickly forming synapses to account for and learn from these new experiences. On the flip side, there's synaptic pruning and cell death. So within the nervous system, there's kind of a principle of use it or lose it, meaning if synapses are not being used or neurons are not being used, they get removed. And this is one way in which the brain maintains efficiency, so it doesn't have wasted connections or wasted cells. Synaptic pruning is, of course, elimination of these synapses that are no longer needed. There's a massive period of pruning that happens between early childhood and puberty, and this accounts perhaps for some of the rapid behavioral changes you might see during this period, and it also accounts for some of the critical and sensitive periods that we're going to talk about a little bit later. Critical periods are this time during which certain functions such as language are particularly easy. So for kids, you might have heard that kids pick up on language more easily than adults, and this is absolutely true. It's in part because their brain is literally physically ready for language development during these times. And actually, if kids are not exposed to language until later in their life, they may not be able to gain full fluency in language abilities. And we'll talk a little bit more about this later. Cell death or apoptosis is happens when neurons are deprived of neurotrophic factor, and this triggers programmed cell death. And this also happens when neurons are no longer needed. So frontal lobe development is rather special. The frontal lobe is the last area of the brain to fully mature, and it continues to develop after 20 years. We're even finding that it might develop into the early 30s. There's one area of the brain, of the frontal cortex, called the dorsolateral prefrontal cortex that develops particularly late. So you can see on this bottom right figure that the red denotes parts of the brain that are less fully mature throughout different time periods. So we have a 5-year-old, preteen, teen, 20-year-old. And so dorsolateral prefrontal cortex is rather immature in a 5-year-old, even in a preteen. It goes more toward green, which is like partially mature as we get to teenage brains. And then even a 20-year-old, it's still this green, light blue color, indicating that it's not fully mature. While most of the rest of the brain is this darker blue and purple color, indicating that it is fully matured. So the dorsolateral prefrontal cortex is responsible for or at least takes part in a number of functions such as working memory, cognitive flexibility, planning, motivation, and attention. Synaptic pruning in this part of the brain continues after 20 years, likely until early 20s in many people. It happens a little bit later in males compared to females. And you can compare the behavior, just that you've observed, of individuals in their early 20s compared to individuals maybe in their early 30s on some of these dorsolateral prefrontal cortex functions. So planning, motivation, attention, things like this, you might see a little bit less maturity in some of these functions in people in their late teens as compared to people in their late 20s. Okay, glial development. So astrocytes and oligodendrocytes start to develop after neurogenesis is finished. And if you remember from our previous chapters, that these two types of glial cells are responsible for the myelination of axons. Myelination is sort of considered to be a crude measure of maturation in the central nervous system because it is one of the last parts of brain development. And different areas of the brain are myelinated at different times. It's been observed that the first places to be myelinated are sort of the basic motor and sensory areas that you need first in development. So when you're young, it makes sense for survival to have at least the basic motor and sensory areas before some of these higher cognitive areas develop.